Hi, I would like to present our work on drug-target interaction prediction using transfer learning. Drug discovery is designed as the process of identifying the roles of bioactive compounds to develop new drugs, and it is usually one of the initial steps in a drug development pipeline. Traditionally, screening is accomplished via low or high throughput experimental bioassays. Since these are time-consuming and costly, machine learning and deep learning methods have been proposed to predict drug-target interactions. This graph shows the number of bioactivities for target proteins for the data in Campbell version 29. As observed, few proteins have many bioactivities, such as more than 1000 bioactivities, while a large number of proteins have few bioactivities, 50 or lower bioactivities, and deep neural network models are hard to train with this low number of bioactivities. This is one of the reasons why we want to pursue this study on low bioactivity data. Deep learning methods. Existing models and architectures for deep learning require large amounts of data. However, recently prediction approaches that can learn from a small number of data have been developed, particularly transfer learning, few-shot learning are among these approaches. Our objective in this study is to design, develop, and implement end-to-end deep learning models using transfer learning for proteins with low or few bioactivity data with the aim of drug-target interaction prediction. 6 protein families are chosen for this study. Similarity results between proteins among 6 families were obtained using Needleman-Wunsch algorithm. It can be seen that almost all families are similar to each other, less than 15%. This is the data filtering pipeline applied on Campbell version 29 to extract training and test datasets. We use kinase protein family as the source family and transporter as the target protein family. Test datasets are independent of the training datasets. We performed controlled computational experiments, and for this, we formed training datasets of smaller sizes going down to 75 bioactivities for the target transporter protein family. Training is achieved with cross-validation, and we used various metrics to assess the model performances. This is the feed-forward deep neural network architecture we used. It consists of an input layer, 3 hidden layers, and an output layer. This is a ligand-based drug-target interaction prediction model. Compounds are represented by their fingerprints. At the output, we have a binary classification which is performed. We train kinase from scratch to use as a source model. This is the first transfer learning model. Here we use the saved kinase model as a source model. All the learned weights are used in initialization of the target model weights. Target model is trained with its own dataset on the already trained target source model. In mode 2, similar to the fine-tuning, the saved kinase model was used as a source model. All the learned weights are used in initialization of the target model weights. However, this time the selected layers are frozen. These layers which means these layers are not updated during the propagation. Only the classification layer is trained with target training dataset. In mode 3, the idea is to use the output of the first n layers of a base network, source network, trained for a source task similar to the target as a generic feature extractor. Instead of the classifier in the last layer, in the source network, a new shallow model is used for the target task. The new shallow classifier can be one of the traditional machine learning models such as random forest, support vector machines, gradient boosting, etc. When these are the graph for the results. When there are more than 100 bioactivity data, target prediction model with shallow classifier works better than the other ones. When there are less than 100 bioactivity data, full fine-tuning achieves better performance. With transfer learning, as we see over here, the convergence is faster, the red lines, and when also the maximum performance is obtained in early epochs. Transfer learning, we would like to proceed with transfer learning with subfamily, sub-subfamily, and single protein multitask learning, and you can get more information in this repository. Thank you for listening.